Hello everybody. Today I discuss a few conclusions about ALS based on the previous 4 presentations. During this presentation, we discuss the following topics. ALS is probably a disease in 2 steps. First, motor neurons are attacked. This can have multiple causes and there are most likely differences between familial and sporadic ALS. Moreover, we deal here with ALS persons with no illness indications. Familial ALS is caused by a germinal mutation in all cells which leads in the offspring to a change of the corresponding protein in motor nerve cells at birth. It is heritable. There is a familial ALS history and is in step 1 easy to determine for the known genes. Sporadic ALS is on the other hand probably caused by a local somatic mutation in stem cells which will form nerve cell supporting cells, developed during life using stem cells. The activation of their corresponding proteins leads to cell death of motor nerve cells with which they communicate. It is not heritable. There is no familial ALS history and is, like cancer, in step 1 difficult to determine. Possible approaches for preventing and delaying ALS are: deactivate mRNA of the active mutated ALS gene using antisense technology, augment the capacity to clean up inactive and wrongly folded proteins, and the long-time application of neuroprotective medication among which certain, like tolampinol, beta-lactam antibiotics, coenzyme Q10 and minocycline, were vainly tested on step 2 ALS patients. Newly proposed treatments in this category are the upregulation of cell stress responses and medication, among which lithium and rapamycin, promoting autophagy and mitochondriogenesis. This however requires the identification and recruitment of familial ALS step 1 persons, and the adaptation of the vision about clinical studies for the prevention and delay of ALS. In the future, the development of a cell scanner may eliminate ALS once and for all. A lot of diseases have in common that they are caused by damaging somatic mutations that slip through the net of the DNA control during the process of cell replacement. It is naive to claim that somatic mutations only point to cancer. Such mutations cause probably also a lot of autoimmune diseases, like the disease of Crohn and Ms, and some neurodegenerative diseases among which ALS and the disease of Alzheimer. The cell scanner should destroy or repair cells with harmful mutations and avoid or stop such diseases. Secondly, a disturbed communication between motor neurons and supporting cells results in faster dying of motor neurons. Motor neurons and supporting cells communicate with each other and they influence each other's gene expression. When too much motor neurons disappear, their communication gets disturbed. The expression of some genes of motor neurons stops, while others are activated. Motor neurons do no longer operate properly in the microglia. The immune system of the nervous system no longer recognize them and destroy them. The attack on motor neurons still continues, but its effect is now negligible compared to the destructive activity of the disturbed communication. There are still a lot of questions about when step 2 begins and gets the better of step 1. Patients in an early disease stage might still be in the transition phase. However, in my opinion, all ALS patients with noticeable paralysis are in step 2 and there are then no longer differences between familial and sporadic ALS. Most people go only to the doctor when their problems become troublesome. In addition, it lasts for sporadic cases, the vast majority, mostly very long before they get their diagnosis. Also, positive results are currently missing, and ALS patients with a recent diagnosis are mostly selected for participation in clinical trials anyway. Patients with visible ALS symptoms in step 2 cannot be cured and will not recover. The motor nerve cells and mainly their connections that are lost will not return or be replaced. Only treatment can bring some comfort. Possible ideas for this are suppressing the destructive activity of microglia, improving the communication between motor neurons and supporting cells, improving the recognition of the own motor neurons, and improving the quality of life by driving devices, such as exoskeletons, speech computers and breathing devices with a brain. Also, the own muscles should be considered. Muscles can after all be regarded as the evolutionary solution for movement, which are activated by neural impulses. Something like this should be tested and fine-tuned in a pilot project. Of course, not all movements can be investigated to evaluate the principle. Manual grasping and twisting is, according to me, a good compromise between big movements and those that require fine motor skills. It could be the focus of a pilot project without bringing the experimental subjects in danger. A neuromuscular performance enhancer device should also be useful for people that have some kind of paralysis. Learn as an ALS patient to live with a deteriorating health. Anticipate, use all available help, have regularly social contact. Make sure that you have a daily, meaningful activity. Focus on what you still can, not on what you no longer can. Be tolerant and patient for yourself and your neighborhood and do not become depressive. Count also on the ALS Liga for help and information. New treatments have to be approved by the European and American drug authorities in order to be usable in Europe and North America. This approval is based on a positive evaluation of clinical trials which test the treatment on humans. However, the current clinical studies compare only sick and healthy participants and guarantee safety and effectivity for the ALS person. This means that you already have to be sick, which is mostly too late for the ALS persons, in order to be recruited in clinical trials. Only the persons having early markers, which are not known yet, can be tested with the protocol of today. Yet this severe regulation contains a crucial limitation. Treatments that may prevent or delay diseases when there are still no visual symptoms cannot be decently tested. Perhaps a similar approach as for the approval of vaccines could be applied, in which only the expected effect is achieved before one is ill, instead of recovery for one that is already ill. We have in any case to lobby for such a protocol that involves genetically charged but not yet sick people in a clinical study. Cooperation will be required to conquer ALS. Not only we have to join for common aspects with other diseases, like cancer, Alzheimer and MS, which have a lot more patients. Physicians should also cooperate and consider assistance from other scientists, like engineers, biologists and biomechanics specialists. Perhaps separate budgets need to be provided for such mutual projects. Testing treatments meant to deal with Step 1 ALS on ALS patients in Step 2 has no sense. Not only will the participants be frustrated by the negative outcome, they will also influence the result negatively and may help to throw a potential treatment for ALS Step 1 in the wastebasket. A new protocol for clinical studies for treatments that prevent or delay diseases when there are still no visual symptoms is however required for the selection of ALS Step 1 persons in clinical studies. For studies with ALS patients, their positive contribution should first be shown in animals with visual illness symptoms, as is also the case with ALS patients. I have of course not the money to pay ALS research projects, but if I could afford it, I would spend my money as follows. First, I would fund research that focus on the development of the cell scanner and the neuromuscular performance enhancer. Furthermore, I would provide money for research in ALS Step 1 on the condition that there is a new protocol for clinical studies for treatments that prevent or delay diseases when there are still no visual symptoms. Currently, These are studies directed to the deactivation of mRNA of the active mutated ALS gene with antisense technology, the augmentation of the capacity to clean up inactive and wrongly folded proteins, and the long-time application of neuroprotective medication. I would also invest in studies dealing with ALS Step 2 if their positive contribution is first shown in animals with visual illness symptoms, as is also the case with ALS patients. Also, the improvement of the independence and life quality of the ALS patient, with the steering of devices with the brain, earns my financial attention. Eliminating ALS is not yet for tomorrow, and there will be for me and other present ALS patients probably no solution. They can only learn to live with their disease and be available for research that makes sense. Still, I see an end in the battle against ALS. The timely and appropriate application of an ALS cell scanner will eliminate ALS once and for all, and although the remaining ALS patients cannot be cured, a performing neuromuscular performance enhancer can improve their independence and quality of life considerably.